Hey guys, today we're going to be doing the ideal cell size lab, and to do that we're going to be using some agar cubes like you see here, and we're going to be using a substance to diffuse into those agar cubes. Today we'll be using vinegar to kind of model and simulate what the ideal cell size would be in an animal or a plant or really anything that's alive and has cells. Here in our agar cubes, you'll notice they are pink. That's because they have an indicator called phenolphthalein in them, which will change color from pink to more clear when in the presence of an acid. Today, our acid is vinegar. You could really use any acid, but vinegar is really safe and also easy to find. So to model this, what we'll be doing is putting our 3 different cell sizes in. You'll see I have a 1 centimeter cube here. I have a 2 centimeter cube, and then a 3 centimeter cube. They feel a little bit like Jell-O, which is why I almost just dropped that. Before we get started, I want you to take a look at your pre-lab questions. Probably go ahead and read your introductory information first too. It'll have some important information. And then do your pre-lab questions, which will require you to do a little bit of math. You might want to pull out a calculator. And when you're done with your pre-lab questions, make sure you come back because we're going to get us started on the procedure. And you guys will get to see what happens when we put these cubes into the vinegar. All right, now that you've finished your pre-lab questions, we're going to follow our procedure. So I have a beaker here. Instead of 400 milliliter beaker, I grabbed one that's a little bit bigger just in case. And I'm going to put all 3 of my cubes in. So first I'm going to throw my 1 centimeter cube in, my 2 centimeter cube, and finally my 3 centimeter cube. All right, so they all fit in there. They're kind of right next to each other. That's fine. They're all going to get covered in the vinegar. So our next step in our procedure is to grab our vinegar, and we're going to pour it in just so that we have enough so it covers all of our cubes. All right, so now all my cubes are in there. I can see almost immediately that the edges start to turn a little bit clear. I'm going to start my timer. And we're going to let it go. Our hope is that we should probably time this for about 15 minutes, but if our small cube gets all the way diffused or clear before that 15 minutes hits, then we'll stop it then. So I'm going to keep an eye on it. You guys can kind of follow along with the video. We won't make you watch for an actual 15 minutes, but you guys can follow along with the video. We'll see how long it takes for that small 1 centimeter cube to diffuse all the way through with the vinegar, or we'll stop it after 15 minutes, whichever one comes first. All right, so we have let our timer run for, I'm going to stop it at 15 minutes and 30 seconds. So that's now. And now what I need to do is take my cubes out of the vinegar so that they stop diffusing. I'm going to take all my cubes out. It's just vinegar, so I don't mind touching it. It's not like a strong chemical. All right. So now what we can see here is we have my 3 cubes, and if I look at them, I notice that they all have kind of this clear box around the outside. So that's all the area that got the vinegar towards it, and that's the amount of space that has been diffused. So what we're going to do is we're going to cut our cubes in half so that we can actually measure how far in the vinegar diffused on each of them. So I'm going to do the best I can with my plastic knife. And I'll flip them so that you can kind of see where the pink and the clear is. There we go. All right. So I have my 3 square centimeters, my 2 square centimeters, and my 1 square centimeter. What I'm going to do is, with my ruler, I'm going to measure how far the vinegar diffused into the edges. Now what I want to keep in mind is I can't just measure one side. I need to measure both because that's how far vinegar diffused total. We want to know the total distance that it diffused. So I'm going to measure both sides. I think I need to pick this up so I can see it better. And let's see. It looks like I have about 0.2 centimeters on each side for my 1 square centimeter. So I know it is 1 centimeter across, and it diffused 0.2 centimeters on the left and 0.2 centimeters on the right, which means out of my full centimeter, I had 0.4 centimeters diffused. That's for my 1 centimeter cube. Now I'm going to look at my 2 centimeter cube. When I measure it, I see again that it went 0.2 centimeters on each side. This time, remember, I have 2 centimeters all the way across, but it went 0.2 centimeters in on the left and 0.2 centimeters in on the right, which means out of my 2 centimeters, 0.4 centimeters got vinegar or were diffused. My last one is my 3 centimeter cube. I'm going to measure. I have 0.2 on the left and 0.2 on the right. So again, I have 0.4 centimeters total that were diffused, but this time it's 0.4 out of 3 total centimeters. What I want you to think about is if this was a cell and let's pretend the vinegar was oxygen or maybe water, which of these cells would be better off? Which of these cells would live better? Let's think about how much of that cell actually got the nutrients that it needed. So when you do your questions, what you're going to be doing is a little bit of calculations first. You're going to be looking at how much of the cell actually got the nutrients that it needed, the vinegar in this case. You're going to be drawing a picture for me and showing me what the cells look like. So take a look at your cells, look at how much is pink, and you'll have to draw it a little bit to scale. And then you'll have a couple of analysis questions. So use the calculations that I read to you. To do your math, you're going to be looking at some surface area to volume ratio between your pre-lab questions and your analysis questions. And you're going to be seeing how the rate of diffusion impacts how much of our cell gets what it needs. And then you guys will be all set. Thanks.